Okay, great. So, welcome everyone to the International Society for the Developmental Origins of Health and Disease Worldwide webinar series for 2022. I'm Nuruddin Mohammed. I'm the secretary for the International Society for the Developmental Origins of Health and Disease. With me, we have Thorsten Braun, and we have the speaker, Professor Sonia Entrina. I would request Thorsten to introduce the speaker, Professor Sonia, and then we take it from there on. Over to you, Thorsten.
Yeah, thank you. Good morning, good afternoon, probably good night, wherever you are, and welcome to this very exciting DOHAT webinar. My name is Thorsten Braun. I'm an obstetrician and maternal fetal specialist here at the Charité University in Berlin, in Germany. And as a board member of the DOHAT Society, I have the pleasure today to introduce you to our prestigious presenter, Professor Dr. Sonja Entringer. Dr. Entringer is a professor at the Department of Medical Psychology at the Charité University Berlin, and also since 2016, associate adjunct professor at the Department of Pediatrics at the University of California, Irvine, United States. She achieved her diploma and later on her doctoral degree in psychobiology at the University Trier in Germany. And between 2007 and 2010, Dr. Entringer did her postdoc at the Department of Psychiatry and Human Behavior at the University of California in Irvine, followed by an assistant and later assistant adjunct professor position at the Department of Pediatrics at the same university. Her main research focus is on developmental origins of health and disease risk, and especially on stress and cellular aging. Her research examines the interplay between maternal placental fetal, neuroendocrine, immune, metabolic, and genetic epigenetic processes on early human development. And her research program has been continuously supported by grants from the European Research Council, the NIH, the German Research Foundation, and other agencies. Dr. Entringer has published over 140 peer-reviewed papers in high-ranking journals such as Nature, and was elected as a fellow by the Academy of Behavioral Medicine Research in 2017. She's a member in several international societies and holds a position in the Scientific Advisory Board of the NIH-funded Telomere Research Network. Dr. Entringer has received several awards, such as the Kurt Richter Award of the International Society for Psychoneuroendocrinology. And so I'm very delightful to have the chance today to listen to Professor Entringer's presentation entitled "The Role of Stress in Developmental Programming of Health and Disease, Risk, Evidence, Issues, and Considerations." For housekeeping, as usual, the talk was pre-recorded to avoid technical problems when you're listening to, but you will have the chance to discuss your questions with her online afterwards. So please stay online for a fruitful discussion afterwards. And I think let's get started and enjoy the talk.
Thank you very much for inviting me and for providing me the opportunity to present our research at this webinar series. What I will try to do today is, after talking about the prevalence of stress and how maternal stress effects can potentially be submitted to the fetus, I present to you some evidence based primarily on our own work that there are indeed long-term effects of maternal stress and stress biology during pregnancy on offspring health-related outcomes and on intracellular processes mediating the long-term effects of prenatal stress on later health outcomes. I will also shortly talk about some of our more recent studies on the effects of maternal preconceptional exposures, and I will then spend some time on going over potential limitations and challenges in this research field and how to address them. The prevalence of psychosocial stress and stress-related symptoms, for example, feelings of anxiety and depression, established that excess chronic stress has effects on mental and physical health outcomes, and that these effects are larger and more enduring when the exposure occurs during the early life stages, especially during the prenatal and early postnatal phase, which is in line with the developmental programming hypothesis. The idea that maternal feelings during pregnancy matter for fetal development is not new. This poster from England from 1919 alludes to it and at the same time provides a potential mechanism for how the effects are transmitted to the fetus. It says worry, fear, and anger may affect the mother's blood, which supplies the food to the unborn baby. It is fascinating to think about this transmission process of maternal experiences to the fetus, because there are no direct neural or vascular connections between the maternal brain that senses and processes the information about stressful conditions and the fetal compartment. All exchange and communication is mediated by biological processes that interface with the placenta. And it is very interesting that the communication between the maternal and the fetal compartment occurs via the same biological systems that, in an already developed individual, mediate the stress response, namely the endocrine, the immune, and vascular processes. It is important to note that these biomarkers of the immune, metabolic, endocrine, autonomic nervous system are essential for fetal development. They play obligatory roles in the initiation, maintenance, and progression of gestation, fetal development, and birth. But they also are responsive to acute and chronic maternal circumstances, including stress and health behaviors, and they mediate the effects of maternal circumstances on fetal physiology by either directly crossing the placenta, for example, a fraction of maternal cortisol can cross the placenta and get into the fetal compartment, or in the case of example, inflammatory ligands that are associated with chronic stress exposure by triggering placental production of inflammation-related biomarkers that are then released into the fetal compartment. And increased concentrations of these different stress-responsive biological ligands in the fetal compartment can permanently alter fetal physiology in organs like the brain, fat tissue, liver, and so on. And these alterations in fetal physiology in turn impact later child developmental and health-related outcomes. To study these processes, we have established several prospective birth cohorts for which we have started recruiting during early pregnancy. And we collect data on maternal stress and related states and circumstances via interviews, questionnaires, and biological samples. At birth, we try to collect placental samples and cord blood, and for most studies, we include a follow-up of the children after birth, in which we focus on two outcomes of interest: brain and neurocognitive development and obesity risk. We have published a series of conceptual and empirical papers showing a link between maternal psychosocial stress and alterations in maternal gestational stress biology, including maternal cortisol levels and offspring adiposity risk and metabolic function. And these effects are already apparent during infancy, they last into adulthood, and we could show that alterations in brain circuits related to energy homeostasis and appetite control play a role in this context. This is one example. In this study, we tested the association between maternal cortisol concentrations during pregnancy and infant obesity risk. Cortisol was assessed at 3 time points across pregnancy in saliva samples collected at multiple times during the day to capture circadian variation in cortisol. Infant body composition was assessed with DEXA scans. After adjusting for key prenatal birth and postnatal covariates, higher maternal cortisol concentrations, particularly during the 3rd trimester, were significantly associated with a greater increase in infant percent body fat from 0 to 6 month age, which is a risk factor for later adiposity and metabolic dysfunction. In a second set of studies, we investigated the association between maternal psychosocial stress, maternal stress biology, and offspring brain development and cognitive function. And this work is mainly led by my colleague Claudia Bus. These studies demonstrate a link between maternal mental state and different kinds of stress exposures on offspring brain anatomy, including the amygdala and overall brain and gray matter volume. Claudia also published several studies on the effects of maternal cortisol and pro-inflammatory cytokines during pregnancy on infant and child brain anatomy and cognitive function. Again, this is an example. Claudia developed protocols to perform MRI scans of the infant brains during natural sleep in infants already at birth. In this study here, maternal cortisol concentrations were associated with a larger amygdala volume, a brain structure that is important for fear processing of 7-year-old children. Furthermore, in this study, we already at birth observed alterations in the way the amygdala is connected to the rest of the brain based on maternal cortisol levels during pregnancy, which in turn predicted symptoms of anxiety and depression at the age of 2 years. We know from decades of research now that numerous conditions during pregnancy have a long-lasting impact on development, health, and disease of the offspring. These conditions influence a diverse set of health outcomes, and the elucidation of mechanisms underlying these effects is an area of intense interest and active investigation. So we put forward the hypothesis that telomere biology may represent a common mechanism underlying the observed effects of different early life adversities on a range of health and disease risk-related phenotypes. Telomeres are non-coding DNA sequences protecting the ends of our chromosomes, and because the DNA polymerase is unable to fully replicate the DNA strand, telomeres lose base pairs with each cell replication, and this over time leads to cellular aging, senescence, and apoptosis. And besides posing an upper limit to the replicative capacity of cells, telomeres are also important for chromosomal stability, integrity of the chromatin structure, and they can regulate gene expression. And because telomeres are so crucial for chromosomal stability and cellular function, it is not surprising that shortened telomeres, mostly measured in immune cells or leukocytes, are associated with a large number of aging-related diseases in humans, as evidenced by more than 50 meta-analyses, including cardiovascular and metabolic diseases and earlier mortality. So why do we think that early life telomere length is important for health and disease risk as opposed to cumulative damage acquired across the lifespan? So for any given individual at any given time point in life, telomere length is a function of the initial or newborn setting of telomere length and of telomere attrition rate over time with age and the counteracting effects of the enzyme telomerase. And there is now considerable variation across individuals in telomere length at birth. Thus, at identical rates of telomere attrition over time, individuals with shorter telomere length at birth will reach cellular senescence earlier, and this will occur even sooner in individuals in whom the initial setting of the expression capacity of telomerase is reduced. And this is why the initial setting of the system seems critically important for susceptibility for aging-related disorders in later life. What is the evidence that maternal stress plays a role here? In adults, there's a lot of evidence that different kinds of stress exposures are associated with shorter telomeres, as recently summarized in this review paper. In vitro studies showed that activity of the enzyme telomerase that counteracts the telomere shortening can be inhibited by biological stress factors like cortisol. Furthermore, inflammatory and oxidative stress processes associated with stress exposure can directly lead to damages in the repetitive DNA structures of the telomeres. And we have conducted a study on the long-term effects of prenatal stress exposure on adult health and disease-related outcomes. It's a retrospective study in about 90 young adults. About half of the study population was exposed to severe prenatal stress as operationalized as major severe life events that happened to the mother while she was pregnant. For example, death of someone close, sudden severe onset of illness of someone close, and so on. The other half was from mothers of unexposed pregnancies. And in these young adults who were exposed to prenatal stress, we found alterations in many physiological systems like the metabolic system, the immune system, the endocrine system or HPA axis function, and cognitive performance that predisposed to disease risk in later life. And in addition, we saw that stress exposure during intrauterine life is associated with shorter telomere length of immune cells in adulthood. And we replicated this effect in a prospective pregnancy cohort in which we showed that the effects of prenatal stress exposure on offspring telomere length are already apparent at birth in cord blood. This association between maternal stress and newborn telomere length has now been replicated by several other research groups in independent study cohorts, and we just submitted a meta-analysis together with Dr. Stacy Drury, the director of the NIH-funded Telomere Research Network, on the effects of maternal stress on offspring telomere length, and we confirmed an overall negative association between maternal stress exposure during pregnancy and offspring telomere length. In one of our prospective pregnancy cohorts, we had data available on maternal pro-inflammatory cytokines assessed at 3 time points during pregnancy. And we observed that a higher pro-inflammatory milieu during pregnancy was associated with shorter offspring telomere length at birth, suggesting that the effects of maternal mental state during pregnancy on offspring telomere length may be mediated by alterations in the maternal placental fetal immune milieu. There's also good news. In a large pregnancy cohort with more than 660 mother-child dietes from Finland, we were able to look at the effects of maternal resilience during pregnancy on offspring telomere length. Resilience can be conceptualized as positive psychological functioning over time in the aftermath of stress or adversity. In this study, we had serial measures of maternal positive affect and stress available, assessed up to 16 times during pregnancy. And based on the positive psychological constructs like positive affectivity, positive mood reactivity, and social support, we built a positivity factor based on principal component analysis. And based on several of the negative mood and stress skills, we computed a stress factor. We then regressed the stress factor on the positive factor, which represents the degree of positivity that is left while facing a certain amount of stress, our concept of resilience in this study. In this large study, we first of all replicated the effect of maternal stress on newborn telomere length, but we also observed an effect of psychological resilience on newborn telomere length. The higher the maternal resilience during pregnancy, the longer the telomeres of the offspring at birth, and this finding on the protective effects of maternal resilience has important implications for potential interventions. In our more recent studies, we become more and more interested in the effects of life circumstances that the mother was exposed to before she became pregnant. Examples are exposure to the mother to adversity during her own childhood, including maltreatment and abuse, and exposure to social disadvantage throughout the life course. The prevalence of childhood abuse and maltreatment, unfortunately, is very, very high, and childhood trauma has lifelong consequences for mental and physical disease risk for the exposed individual. Recent studies suggest that these long-term consequences are not restricted to the exposed individual alone, but they also may get transmitted to their children, who also present with a higher prevalence of psychiatric disorders and obesity risk, for example. The pathways of such intergenerational transmission are believed to occur during the offspring's childhood as a consequence of compromised parenting and caregiving in mothers who have been exposed to childhood trauma. What we have proposed is that the transmission of these effects may start even earlier, during the child's period of intrauterine development. Offspring of mothers exposed to childhood adverse experience may receive cues about maternal early life conditions via alterations in stress biology that are carried forward from the non-pregnant state into her pregnancy. And these alterations in maternal stress biology during pregnancy may in turn influence the developmental trajectory of the fetal brain and have long-term implications for his or her mental health and disease risk. Indeed, we and others showed that maternal childhood trauma is associated with alterations in stress physiology during pregnancy, including higher cortisol concentrations and higher inflammation. And my colleague Nora Mork showed that brains of babies whose mothers were exposed to trauma in their own childhood were smaller in volume due to a global difference in cortical gray matter compared to babies of non-exposed mothers. And we saw these alterations already at birth, when at a time point when no postnatal exposures could have had an effect. In several independent studies, we find that factors related to social disadvantage are associated with higher inflammation during pregnancy, For instance, in this study, we used the MacArthur Scale of Subjective Social Status to let the participants indicate their social standing with reference to their community and to the nation. And we found that lower perceived subjective social standing was associated with a higher pro-inflammatory state during pregnancy. And in this birth cohort study established in Berlin that was enriched for Turkish immigrant women, we found higher concentrations of pro-inflammatory cytokines in migrant pregnant persons compared to non-migrant pregnant individuals. Now I'm going to mention a few issues, considerations, and challenges when doing this kind of research in humans. It is important to note that the effect of any given exposure on biological pathways and outcomes is not independent, but is conditional upon other exposures or states. In other words, stress, for example, does not occur in a vacuum. There are many examples of how our nutritional state can impact our stress response and vice versa. We also know that stress can impact our immune response to an infection and so on. So it is difficult to study these complex interactive processes in human observational studies. The effects of biological transmission pathways or systems are also not independent, but are conditional upon the states of other biological systems. Perturbations in one system, for example, the HPA axis, lead to secondary perturbations in other systems, for example, the immune system. And individual differences in the magnitude and duration of perturbations in any given system are in part a function of the state of the other systems. In most of these types of studies, we are very limited in terms of the number of biosamples that we can collect during pregnancy. And with just a handful of samples collected across gestation, it is not possible to capture within person variability over time appropriately, which is very important, for example, in the context of the effects of variation in stress or cortisol on pathophysiological outcomes. Often in these studies, we get blood samples and quantify circulating biomarkers, for example, concentrations of pro-inflammatory cytokines. Circulating biomarkers, however, or circulating concentrations of cytokines may not adequately capture the functional capacity or characteristics of a system. For example, concentrations of IL-6 may reflect a primary response to a stimulus or a secondary compensatory response to the initial stimulus. To adequately test the functional capacity of a system, we would have to rely on experimental approaches that evoke and quantify a response to physiologically relevant challenges, for example, like we do with the glucose challenge test. And of course, it is ethically not possible to randomize pregnant women to stressful conditions. So we have to be careful with drawing any conclusions about causations with these types of studies. To address the first point about stress effects being conditional upon other exposures, we published several conceptual papers on the interplay between maternal stress and nutrition during pregnancy. And in an empirical paper, we showed that the effects of a pro-inflammatory diet during pregnancy on concentrations of inflammatory cytokines are conditional upon the effects of perceived stress. In other words, the higher the maternal stress levels the mother reported, the higher the association between pro-inflammatory diet and inflammatory cytokines during pregnancy. To address the issue of within-person variability of stress over time, we established ecological momentary assessment protocols during pregnancy. Over a 4-day period in early, mid, and late pregnancy, women answer a mobile phone-based electronic diary several times a day to assess stress and mood states in their natural environments. They wear a device to collect cardiac and physical activity, and this allows us to capture within-person variability of stress and physiological responses to it over time. Furthermore, they collect 5 cortisol samples per day over these 4-day periods during each pregnancy trimester, which results in about 60 samples per woman across pregnancy. And although in experimental settings in which you expose individuals to stress, for example, in a lab, an increase in cortisol levels has reliably been shown, many studies fail to show a link between stress assessed with traditional stress questionnaires and, for example, cortisol levels. In this EMA study of 152 pregnant women, we used linear mixed models to examine associations between psychological stress and cortisol assessed concurrently at the between and the within person level. And after accounting for the effects of key determinants of variation in cortisol, we see significant associations between momentary stress and cortisol levels, however, only if we look at this at the within person level. No association was evident for traditional retrospective measures of stress with cortisol at either the between or the within person level. These next 2 points regarding the interplay of different biological systems and the importance of characterization of the functional capacity were addressed in one study. We used a challenge test that combines two systems, the immune system and the HPA axis. We applied an ex vivo stimulation assay, a dynamic measure of immune function at the cellular level, to investigate pro-inflammatory IL-6 responsivity of leukocytes to an antigen, in this case LPS, and the regulation or dampening of this pro-inflammatory response by glucocorticoids, which gives you a measure of glucocorticoid receptor sensitivity or resistance. Across 2 time points in pregnancy, Black women exhibited significantly higher LPS-stimulated IL-6 release and reduced glucocorticoid regulation of the IL-6 response, which suggests higher glucocorticoid receptor resistance relative to white women. There was no difference in the concentrations of circulating IL-6 levels between Black and white women in this study. And this glucocorticoid receptor resistance has implications for the risk of preterm birth, as we could show in a different study. Regarding this last point that addresses the issue of causality, what some groups have done is they took advantage of so-called natural experiments, for example, exposure to natural disasters during pregnancy. In a study we conducted together with Thorsten Braun at the Charité in Berlin, we took advantage of the fact that antenatal treatment with synthetic glucocorticoids of individuals in danger of preterm labor is common clinical practice. Thorsten has established a cohort of women exposed to the synthetic glucocorticoid betamethasone during pregnancy and non-exposed women as a comparison group. We measured placental DNA methylation at birth, and the findings are not confounded by preterm birth because the groups were matched for gestational age at birth. Exposure to betamethasone was significantly associated with lower placental DNA methylation as an enhancer of the gene coding for FKBP5. And FKBP5 is important for the regulation of the biological stress response. It modulates glucocorticoid receptor activity, for example, of immune cells. In another pregnancy cohort, we saw that lower DNA methylation at this enhancer site was associated with higher expression of FKBP5 in the placenta and a co-expressed gene module. And this module is enriched for genes associated with preeclampsia and involved in inflammation and immune response. So this very small epigenetic alteration in a gene associated with the stress response may therefore result in a higher pro-inflammatory milieu during pregnancy, which in turn has implications for fetal development. To conclude, there is evidence from several independent studies that maternal stress and gestational stress biology is associated with offspring disease-related outcomes and underlying cellular mechanisms. Our studies also highlight the importance of preconceptional events and life circumstances related to stress exposure of the mother and social determinants of health. Future studies should initiate biological data collection in stress intervention trials during pregnancy, and we are currently working on implementing so-called micro-randomized trials or just-in-time adaptive interventions, in which one randomly assigns a stress intervention when the woman is in a high stress state in an ambulatory setting, and we can then look at the effects on, for example, stress biology during pregnancy. Future studies should also include measures on psychological stress and biological samples of the father. And the ultimate goal should be to develop screening tools for early detection of vulnerability and previous adverse exposures, to detect very early in life who is at heightened risk and therefore may benefit from behavioral or social interventions. I would like to thank the organizers again and Torsten for inviting me to this webinar series. I would like to thank my collaborators and the funding agencies who have supported these studies, and you for your attention. Thank you very much.
Yeah, all right, sorry. So thank you, Professor Sonia, for an excellent presentation. Any questions for Professor Sonia? No questions so far from the audience. Okay, so I have one question for you, Professor Sonia, and that is, did you take into account the low birth weight neonates? Because we know low birth weight neonates or growth restricted fetuses, they do tend to show smaller brain volume and eventually altered neurodevelopmental behavior as early as at the neonatal stage. That's been proven. So did you take that factor into account?
So in most studies, we exclude babies with very low birth weight to exactly account for this effect. And mostly we include birth weight as a continuous variable, as a covariate birth weight, as well as gestational age at birth, because yeah, you are absolutely right. These variables themselves have effects on brain development, on obesity risk, and so on. So that's why either by excluding them by design or by controlling for them, we adjust for this, yeah.
And again, the same the question along the line for gestational diabetes, diabetes in pregnancy. So
Yeah.
you also ruled out in the study design, I understand, women.
So yeah, we mostly recruit so far in our studies healthy pregnant women. Of course, some of them develop gestational diabetes over pregnancy, and in most of our studies we have like a handful or 10 pregnant women. And what we did sensitivity analysis. So we ran the models with and without these women, and because it was so few of them, we mostly confirmed our effects. An interesting study to do would be to look at the interactive effects of stress and gestational diabetes on infant outcomes, because the endocrine system and the metabolic system interact so much, especially during pregnancy. So that would be a project, a new project.
Yeah, thank you very much, Sonia, for this great talk. And again, to our listeners, 26 people are online at the moment. Please use the chat function if you have any questions, and we will read the questions out to Sonia. And there was just one question coming up. And I'm just read it. You mentioned in one of the slides that you started small intervention trials testing effects of strategies to reduce stress during pregnancy. What sort of interventions are you considering? For example, mediation, diet, medication?
So in these studies, what we want to set up is we want to sense when the woman is stressed during her everyday life with the sensors and this diaries that I showed before. And when the woman is stressed, we want to send her a signal to engage in a stress reduction intervention. For example, what has been shown to be very effective in terms of bringing the autonomic nervous system down is a breathing exercise. Taking 6 deep breaths, slowing down your breathing has huge effects on autonomic function. So this is something that I think will be effective, but the woman has to be reminded to do these interventions in a state when she needs the intervention most. And mostly when we're stressed, we're busy with other things, we don't think about intervening right away. And that's what we want to try to get at with these studies. So we want to develop a system that passively senses stress based on autonomic nervous system measures, then send a prompt to the woman to, for example, try to go to a quiet space and to engage in breathing exercises, or also to do some kind of cognitive restructuring of the situation. So to ask her to take a step back and to think about the situation. Is it really as threatening as she perceived it in the first place? So these are kind of micro interventions that you can try to build into everyday life, and you can kind of individualize them, because I think for stress intervention, there is no one fits all solution. So some people need this kind of interventions, some need others, and you need them in special situations. It doesn't help you much to breathe on your weekend when you have time, but not in the situations where you actually need it. So yeah, that's what we are trying to develop.
Very nice. To take this up, you nicely showed that with a positive intervention, you can influence the telomere length at birth. To pick up this topic with telomere length, which I think is very fascinating, and probably not everybody from the audience is familiar with this. When actually is the telomere length fixed during pregnancy? So what time of pregnancy is it determined? Is it really at birth, or is it a continuous development until birth?
So you inherit telomeres from your parents, and that's the first telomeres that you have after fertilization. And as I explained, with every cell division, your telomeres get a little bit shorter. However, during early development, your cells express a lot of telomerase because of this growth and cell division to kind of keep up with that. So depending on how long your telomeres are at conception, really, not at birth, and how much telomerase you're producing, telomere length gets set at birth based on these factors. And yeah, as I explained, oxidative stress, inflammation, and so on can not only during later life, but already during intrauterine life have an impact on telomere length. And in animal studies, they could show that, for example, when you treat the pregnant dams with TNF-alpha, with the pro-inflammatory cytokines, this has direct effects on the offspring telomere length during fetal development. So there is a certain telomere length that you inherit from your parents, but then the effects could be on telomeres, could be through all these environmental, stress-related, nutrition-related factors.
Okay, cool. And how do you think is the telomere length during pregnancy affected by pregnancy issues, like as you said, with maternal stress, diabetes, birth, growth restriction, etc. So how is the mechanistic pathway to reduce the telomere length during pregnancy?
So, for example, with cortisol, maternal cortisol can pass through the placenta into the fetal circulation, through the glucocorticoid receptors in the fetal compartment can have effects also on fetal development. And we know that if you incubate, for example, immune cells with cortisol, this inhibits the activity of the enzyme telomerase, which counteracts the telomere shortening. So that's one possibility of a mechanism how cortisol, for example, can make our telomeres shorter. And then oxidative stress-related processes can directly damage the telomeres, the repetitive DNA sequences of the telomeres. And oxidative stress is something that is related not only to psychosocial stress, but also to inflammatory or diets that are high in pro-inflammatory potential, in unhealthy lifestyles and so on. So all of these factors produce higher oxidative stress in the mother that can induce processes in the placenta that then impact oxidative stress related processes in the fetus that then can have effect on the fetal telomeres.
Cool. There's another question in the chat. Thank you for this great talk. What kind of paternal biological measures would you advise for future studies in this area?
So, as I explained earlier in my talk, we're getting more and more interested in these preconceptional exposures. And we know that they can have long-term effects, or events that happen, for example, during childhood can have long-term effects on the individual in terms of his or her physiology, her disease vulnerability, and so on. And I explained how this works through maternal exposures on fetal programming, but of course the same can happen to a father. And there are animal studies that show that early life exposures have epigenetic effects on sperm cells that then can inherit it to the offspring. So it would be very interesting to not only get data on maternal preconceptional exposures, but also paternal preconceptional exposures in terms of traumatic experiences, stressful life events, and so on. Some studies, some cohorts that I know of already collect sperm samples of the father, but then you have to do it at the, if you are interested in defects on the child, you have to do it at the same time or around conception, which might impose some challenges logistically. And also more and more studies become interested in the interaction between the mother and the father, which of course can have a preconceptional impact, but also an impact during pregnancy and also once the child is born. So these are a few examples of variables that we're trying to get in our new studies from the father. I don't think it's very useful to collect stress hormones of the father post-conceptually or after the baby is born. This could give you some hints on his stress level once the baby is born, but it doesn't tell you much about previous exposures. But yeah, getting sperm samples would be really interesting.
Cool. Can you speculate the effect of the father on telomere length and long-term development? Do you think it's equal to the mother's effect size, or do you think it's probably even bigger or greater?
That's very hard to say because it works through several pathways, because already the telomere of the father that he inherits to the child could be shorter based on his own stressful experiences. Then if the father is highly traumatized, his behavior towards the mother might be different. If he has experienced violence as a child, he might have different interactions with his partner or wife, and for that could impose more stress on the mother, which then of course could impact maternal stress physiology during pregnancy, which then of course could have an effect on fetal development, and this could go on postpartum after the child has been born. So having a child is in some instances very stressful experience, especially if you have experienced stress in your own childhood, and then through the interaction with the mother or through the direct interaction with the child, this again is another transmission pathway through which these early experiences of the father could be transmitted to the developing child, her brain, his or her brain, and so on.
Okay. Great. So we have one more question out here. Is there a role of gut microbiome and the influence of it on stress during the fetal development?
Well, yes, studies have shown that. Studies have measured maternal cortisol during pregnancy and showed that there are effects on the microbiome already at birth. And I think, yeah, there's a disbalance of some bacteria. I'm not very familiar with the microbiome, but these studies are out there. And in our studies here at the Charité, with Thorsten Braun, we are collecting maternal and infant stool samples. So we can, in future analysis, look at that in our own studies. But I would assume there are effects, yes. Because in developed adult, there's a lot of crosstalk between the brain and the gut microbiome.
Okay, and just one more question coming up. Is there a role of maternal age?
Yes, maternal age plays, of course, a role on her own telomeres. The older we become, the shorter the telomeres. So the initial telomere length that is inherited to the fetus might differ or vary based on maternal age. Interestingly, there is the opposite relationship between fetal telomere length and paternal age. So it's the paternal age effect, which is controversially discussed. The older the father at conception, the longer the telomeres of the offspring. And this is in part explained with the effects of telomerase, because telomerase is very active in sperm cells to keep them alive and going. And the older the father, the more active potentially the telomerase in the sperm cells, and that could explain the paternal age effect on offspring telomere length. Yeah, that's out there in the literature. Nobody really can explain it, but it's opposite to the maternal age effect on telomeres.
Okay.
Maybe, Sonia, you can also explore a little bit more on the sex differences we see in so many models with respect to programming, and the changes you see in telomere length in your studies.
Yeah, so what you see in most cross-sectional studies in adults is that telomeres are longer in women than in men, and this is mostly due, or people think it's due to the effects of estrogen on telomerase, because it has protective effects of telomerase. It can increase its activity, and therefore the telomere shortening is less in women and in men. And the difference between men and women in telomere length gets shorter after menopause, which is another hint at this. We also observe in our newborns that telomeres are longer in female newborns than male newborns. However, the effects of maternal intrauterine exposures on offspring telomere length are similar for males and females in our studies. So we don't see differential effects of maternal stress exposure on male or female fetuses in terms of the telomeres. At least.
Thanks.
There's one more question that has come up here, is whether you have tested interaction effects between telomeres and the postnatal environment, and whether they indicate evidence for diathesis stress.
For which kind of stress? Or, oh, yeah. So, of course, maternal stress during pregnancy and postnatal stress exposure are not independent. So if the mother lives in a stressful environment, if she undergoes stressful circumstances, life events, and so on, these effects don't disappear after birth. And there are models like the diathesis stress model that suggest that these early life exposures make us more vulnerable to stress effects in later life. So it increases our vulnerability to react in a certain way to stress exposure after birth. And this could be an advantage in stressful environments, that we are more vigilant and attentive to stressful cues. So in a stressful environment, this could be an advantage, but it could also make us more prone to psychiatric diseases like anxiety disorders and so on. And yeah, this would be in line with the diathesis stress model, that these exposures make us more vulnerable to the effects of exposures later in life, like a second hit model.
Okay. So another question for you, Sonia. Do you think antioxidant diet could be a good intervention to maternal stress-induced programming?
Well, yeah, I mean, in terms of telomeres and telomerase activity, there are a lot of studies that look at the effects of changes in lifestyle, and of course, dietary interventions are a big part of it, and especially antioxidant rich diets have been explored in these studies. The effects that I've seen so far are small, but mechanistically, I think they should be there, and we need larger, better controlled studies to really show them. And I'm not aware of any study that either looked at the effects of antioxidant diets during pregnancy on offspring telomere length, or that looked at whether the effects of any kind of intrauterine exposures could be compensated by an antioxidant diet that you would actually feed to the child after birth. But it's a promising intervention, I think, potentially.
Okay, and one more question for you, Sonia, is whether maternal stress has any impact on the cardiovascular system of the offspring?
Yes.
Negative influence.
Not only do I think so, but there are studies that showed large effects of maternal stress on higher risk for cardiovascular diseases in later life. Large human observational studies could show this, but also animal models that looked at the mechanistic underlyings of these effects. Yeah, so they're definitely there.
All right. So no more questions for you at the moment. And thank you, Sonia, for an excellent talk. Thank you for being there for us. And thank you for accepting our invitation to be here for the DOHA Worldwide Webinar Seminar. Thank you, Thorsten, for being here. And I believe with that, we conclude our today's seminar. We look forward to welcoming you again in November next month for another exciting talk. So stay tuned. And for those who are not yet the member for the International Society for the DOHA, please feel free to surf around with our new website, which has already been launched a couple of days back. And we hope to see you soon next month again.
And thanks again
thank you
for
so
the
much.
invitation. Yeah, it was a
Thank you
pleasure
very
and my
much
honor
to
to be here.
everybody.
Thanks.
Thank you.
Thank you. Bye-bye.